Okay, so welcome to this next video in which we're discussing type 2 activation of endothelial cells. Okay, so far we've seen that later on in type 2 activation, what's going to happen is that we're going to stop expressing E-selectin and CXCL8, and you're going to replace E-selectin with this intercellular adhesion molecule 1, ICAM1, and also with this vascular cell adhesion molecule 1, VCAM1. In addition, you're going to replace CXCL8 or interleukinate with this CC chemokine ligand 2, also referred to as CCL2. Now, this is going to mean that the target for these endothelial cells is going to change from neutrophils to monocytes. Okay? So what happens is that monocytes have receptors for ICAM1 and VCAM1 on their surface. Okay, so these will bind, and really it's only necessary for one of them to bind to get the monocyte tethered, but two of them could bind potentially. So the ligand for VCAM1 is what's known as VLA4. Okay, so VLA4 stands for very late antigen 4. Okay, and then the ligand for ICAM1 here is what's known as LFA1, so LIFA1. Okay, right, so LIFA1 binds to ICAM1, and also VLA4 binds to VCAM1. So I'll colour VLA4 in turquoise here. Okay, and that tethers the monocyte to the endothelial cell layer. Now again, you then have a tight adhesion, which is formed by the interaction of a CC receptor, okay, so CC chemokine receptor with the CC chemokine ligand 2, the CCL2. So this that I've drawn here is a CC chemokine receptor. Okay, and those are often abbreviated to CCRs. Okay, so this is a CCR on the surface of our monocyte here. Okay, right, so let me just complete that arrow. So we'll colour in the CCR in orange. Okay, right, so then what you've got is a tight adhesion between the monocyte and these molecules on the surface of the endothelial cell. What will now happen is the monocyte will undergo diapedesis. It will move between the gap between two neighbouring endothelial cells into the interstitial fluid, and when it gets to the interstitial fluid it will undergo differentiation into a macrophage, and now it will phagocytose the Staphylococcus aureus that is in the interstitial fluid. So this is how you get the movement of monocytes out of the bloodstream into the affected area, and this will be occurring across the endothelium of the capillaries and the venules. So this is known as monocyte extravasation. Okay, so extravasation. Right, so it's an important change that occurs in type 2 activation as you progress, basically. So instead of recruiting neutrophils, you switch to recruiting monocytes, which will differentiate into macrophages. So initially you bring in the pawns, you just help type 1 activation, which is also recruiting neutrophils, and then later on you switch. Type 2 activation switches, so it turns off the E-selectin, it turns off the CXC chemokine ligand 8, and it replaces it with these ICAM1 and VCAM1 and the CC chemokine ligand 2, which instead of targeting neutrophils, targets monocytes, which will diapedese across the endothelium and differentiate into macrophages. Okay, so that's it for this video.